So we have 2 more lectures to go, and then I actually like better the last 2 lectures than the first 2 lectures. But you can tell me tomorrow how you feel. But I like the last lectures better because in the first 2 lectures I told you about kind of our classical work, classical in the kind of sense of. Not that we're classics, but rather that this is the well-established methods to study the function of proteins using gene knockouts. And in last years, my lab kind of started a new direction where we try to use the modern mass spectrometry approaches and then proteomic, proteome-wide and genome-wide analysis, and to use mice to understand the function of the proteins in a kind of a global way. We actually called it genetic. We published a paper in Nature that I'll tell you a little bit like tomorrow, and we call this approach a genetic proteomic because this combines the genetic engineering of mice with the analysis of the whole proteome. And today's lecture is going to be the first of this kind of a new proteomic approaches, and then the last and the second lecture tomorrow, right? Tomorrow's Friday, right? Is going to be to be continuation of this. This one is going to be about cyclin E, and I mentioned to you that cyclin E is a cyclin that is induced during the S phase entry, and it's narrowly peaks at the S phase, and therefore you can use cyclin E as a marker of the S phase cells. You can take the cells in a dish and you can stain them for cyclin E. This is, by the way, not our work. It says copyright UpCam. So I'm not violating, hopefully, any copyright. I'm just advertising UpCam antibodies, which we actually don't use, but this is just staining for cyclin E, and you can see this nucleus is green and this nucleus is green. Why? Because these cells are in early S phase, whereas these cells are blue. The blue is kind of counter-staining for DAPI. They are not in early S phase. If you take the cells in a synchronous way and then you stimulate them, they enter the cell cycle, and then you immunoprecipitate cyclin E and do in vitro kinase assay to look at the kinase activity of cyclin E, you can see that early G1 phase there is very little kinase activity, then S phase there is a kinase activity, and then this activity goes away. And the same in cancer cells. If you take cancers and look for cyclin E is expressed and is expressed in the cells that are in an S phase. And this is the in situ hybridization, where we take the embryos and stain them for cyclin E1 and E2. The red is a positive, and you can see that all proliferating cells express cyclin E1 and cyclin E2. And this is a northern blotting. This is cyclin E1, and this is cyclin E2 from adult organs. And as you can see, if you take, for example, lung or liver or kidney, which doesn't contain proliferating cells, there is no cyclin E1 or cyclin E2. When you take testes, where there are proliferating cells, or if you take thymus, then there's high level of cyclin E1 and cyclin E2. So this is consistent with the fact that cyclin E is a component of the cell cycle machinery. But 13 years ago, Yang Geng in my lab noticed that in the brain there is high level of cyclin E1 and E2, although this is a non-proliferating compartment. And independently of this, the group of Miyamiya in Japan has showed the same thing, that in a quiescent brain there is high level of cyclin E1. This is Junko Dajima. Junko Dajima comes from Japan. She came from Osaka. She was a postdoc in the lab, and she decided to try to solve this mystery, why there is high level of cyclin E in the brain. So when she started, first of all, she did a Western blotting, because all work done so far was at RNA level, and she shown that cyclin E is high in the embryonic brain, which is not surprising. This is a proliferating brain, but also an adult brain. Whereas, for example, in lung or kidney or heart, when there is little proliferation, there is no cyclin E. And then she dissected different structures from the brain, including striatum, cortex, hippocampus, and she found that cyclin E is expressed everywhere. She then did immunostaining. She cut the sections. This is actually hippocampus, where our memories are stored, or cortex. This is thalamus. This is staining for cyclin E. This is staining for nu N. Nu N is the marker of terminally differentiated neurons, and this is the colocalization of these two, showing that truly cyclin E is expressed in the terminally differentiated neurons, and is not expressed in glial cells as shown here. This is staining for the hippocampus. This is cyclin E, very nicely stained hippocampus. This is GFAP. This is for glial cells, so there is no colocalization between cyclin E. Either you have GFAP positive, meaning either you are glial cells, or you express cyclin E, and then you could say, how do I know that this is cyclin E? Maybe this guy is showing me some artifact. So this is cyclin E knockout brain, and you can see that now this is gone. So this is a good antibody is working. So the first thing, so we have a pathway like how to find out what cyclin E is doing in a brain. And the first experiment Junko did is that she took neurons, she cultured them, and then she did confocal staining for cyclin E. You cannot see it very well here, but maybe better here, where you can see that cyclin E is expressed in cell soma and dendrites and neurites. So cyclin E is expressed everywhere. And this is different than in proliferating cells, because in proliferating cells cyclin E is strictly nuclear. Whereas here in terminally differentiated neurons, cyclin E is present all over the cell. And this suggested that cyclin E might play a different function in neurons than the textbook function in the cell cycle. So how can we find out what's the function of cyclin E in those neurons? So a technique that we use and many people use is to ask, what are the binding partners of our protein, whom our protein is binding? That's quite often how you answer about the function of the protein. And the way we often do, and I'm sure you do it too, is that we take the cells and then we express our protein, let's say cyclin E, but we attach two amino acid sequences coding tags. One is, for example, flag tag, and the second hemagglutinin HA tag. These are short amino acid sequences against which could be also MIC tag, because they are very good antibodies against those tags. So now you take the cells you lyse, and then you pass it through the column which contains anti-flag antibodies. So theoretically all proteins are going to flow through, only your protein is going to be bound because you put flag tag, this short sequence, at the end of your protein, so you capture your protein together with associated proteins. The truth is that you also capture a lot of junk. So when you elute your complex, you put it onto the second column, which contains anti-hemagglutinin, anti-HA antibody. So then again, hopefully junk falls through, your protein is captured because you also put a HA tag, and now you elute a hopefully very pure complex. You can run it on a silver stained gel. This is the control, where this is only junk, and this is where this is your proteins. Every band here corresponds to a different protein, and either you can cut out, you can say, okay, well, I want to know what is this protein, what is that protein, or as we do it more routinely now, we just take everything and we do the shotgun mass spec sequence, so we just sequence everything. This is great if you want to know what is the function of cyclin E in cultured cells, but we are not interested in cultured cells. We wanted a brain. We wanted to know the function of cyclin E in a brain of a mouse. And to do this, we started with a new approach called genetic proteomic approach, where Junko made a knock-in strain of mice. And it's very important that you understand this slide, because if you understand this slide, that you understand everything else. And the way we do it is that we go to the single cell, and rather than knocking out, rather than removing the gene, or I told you yesterday about the situation where we put a point mutation into the gene, now we are inserting short DNA sequences encoding those tags, flag and HA tags. So we are making the new gene, which is exactly like the old one, but there is sequences encoding flag and HA tags. So this mouse, when we brought to homozygosity, in every cell is going to express cyclin E, of course. It's driven by its normal promoter, so everything is physiological, except that there are two little tags attached to this protein. Do you understand it? Yeah? So we are attaching those tags because now we can take any organ and we can put it through those two columns, and we can purify our protein together with associated proteins. So this is the approach where now we have mice running around, where we can take any compartment, I think it's your next slide. We can take any organ, we can lyse it, we can put it through the first column with anti-flag antibody, then the second HA antibody, and now we have a pure complex, and we can do mass spectrometry. So we can analyze, and then we do mass spectrometry by doing it over and over and over and over again until we see no new proteins. So this way we can have kind of the full the full coverage of the interactors, and we can see what are the interactors of our protein in this organ versus that organ, or we can see what are the interactors in breast versus breast cancer, how does it change during cancer progression. But in this case, we wanted to know for the brain. So we take brains from the embryos, we take 20 embryos per experiment, then we put through those two columns, and now we have a complex of cyclin E from the embryonic brain. This is cyclin E, and these are interactors. Or we take adult brain, and then we purify the complex. But we don't want to have two lists. One is we want to know what's different, right? Because we want to know what's the difference between the embryonic brain and adult brain. And the way we do it is that we purify the complex from embryonic brain, and this is in one tube, and we purify the complex in adult brain, and this is in the second tube, and then we take those proteins, we digest them into peptides, and then we label them with different isotopes. This tube with one isotope, that tube with another isotope. This technique is called ITRAC, or this is quantitative proteomics. So now we can mix them, and we can do mass spec together. But the mass spec for every peptide that the mass spec is seeing is going to tell us whether it contains one isotope or the second. So we can have the ratio of the abundance of every peptide between adult brain versus embryonic brain. So this way we can quantitatively compare, not just having a list and saying, oh, this is different, but rather we can see how much is labeled with one isotope versus the second isotope in cyclin E binding proteins. And I think it's shown here. So we take the cyclin E associated proteins from embryonic brain, we label with one isotope, from adult brain another isotope, mix, do mass spec together, and then in the end we are going to look at the ratio. So if a given protein is binding cyclin E in adult brain and embryonic brain, the ratio is going to be 1. So we are not going to be interested in this. If something is going to be more abundant in the embryonic brain, well, we're not interested, because in embryonic brain we think we know what's going on. This is a proliferating compartment. We want to see what's specific to adult brain. What is the pattern of cyclin E in adult brain? So this is the result. So most of the interactors, so this is the number of proteins, and most of them have the same ratio, meaning they're binding cyclin E and embryonic and adult brain. Some of them are only found or mostly found in embryonic brain, proliferating brain, and these are our known cell cycle partners. But we are interested in these proteins. These are the proteins that are binding cyclin E in adult brain. There are only two that were enriched. One of them is cell cycle inhibitor P27, and the second cyclin-independent kinase CDK5. So at the first kind of approximation, when you see that cyclin is binding cyclin-independent kinase, it doesn't sound like kind of a shocking discovery. However, the name of this protein is actually incorrect. Although it's called cyclin-independent kinase, it does not bind bind cyclins. It binds the proteins called P35 and P39, which are non-cyclin proteins. It is required, together with CDK5, it's not expressed in proliferating cells, it is expressed in differentiating cells, and it plays a key role in neuronal differentiation. So first of all, we verified what we found by mass spec. So here we are immunoprecipitating cyclin E from the embryo, and we found that it binds CDK2, a textbook partner of cyclin E, but very little CDK5. In adult brain, it does not bind the textbook partner CDK2, but now it binds this new partner, which is CDK5. And I also told you that the second protein that it binds in adult, but not in embryo, is a P27 cell cycle inhibitor. In fact, we found that there's a ternary complex, cyclin E, CDK5, and P27, because we can immunoprecipitate cyclin E, re-immunoprecipitate CDK5, and then Western blot for P27, and we have this complex. So now we are doing a developmental time course. I told you talking about the other experiment, that in mice the pregnancy is 19 days. So now we are going to sacrifice embryos at day 11, 14, 16, and P0 meaning postnatal. We are going to take the brains and we are going to look at the level of cyclin E. So the level of cyclin E in developing embryo is very high. That's not surprising. There is rapid proliferation, so you would expect high levels. Then when the mice are born, at P0, the level of cyclin E goes down. Again, no surprise here, right? The cells stop proliferating. But now, look what happens. Now cyclin E becomes induced. And this is no proliferation there. This is where neurons are doing something that is not dependent, that has nothing to do with the proliferation. This is CDK2, the cell cycle classical textbook partner of cyclin E, is highly expressed in proliferating cells, and then is extinguished after birth. No surprise, because after birth there is no proliferation. But the second partner, this new partner of cyclin E, CDK5, is not expressed here because now the cells are proliferating, but now is induced afterwards, and now it binds cyclin E and does something. So first of all, we ask, okay, well, if we mix cyclin E together with CDK5, can cyclin E activate the kinase activity of the CDK5? So let's look. CDK2 on its own, this is a cell cycle classical partner of cyclin E, has no kinase activity. If you put cyclin E, it's activated, so our cyclin E is okay. CDK5 on its own typically has no kinase activity. If you put a textbook partner, P35, there is a high kinase activity, but when you put cyclin E CDK5, there is no kinase activity. So cyclin E can bind CDK5, but cannot activate kinase activity. So we are really puzzled. If it were to activate kinase activity, it would be great. That would be kind of easy way out. So we found kind of that cyclin E can bind CDK5. And actually, there is a person who, called Andrea Mustaccio in Italy who solved the crystal structure of P35 and CDK5 and found how P35 activates CDK5. And I called him. Actually, I called his assistant, Marina Mappelli, and I told her, we are trying to make this complex, and we cannot find any kinase activity. And she said, don't bother, you will never find any kinase activity, because she said, when we do the crystal structure of this protein, we found that P35 uses a completely different molecular mechanism to activate CDK5, which is completely different than, for example, cyclin E is using to activate kinase. So they said that maybe it's binding, we believe you, but it's not going to activate kinase activity. And indeed, we could never ever see any kinase activity. So we have to come up with something, right? So we proposed that maybe there are two kind of complexes: the active textbook complex, which is P35, this no activator of CDK5 together with CDK5, and then inactive complex with cyclin E, CDK5, and P27. And then maybe we hypothesize cyclin E acts as inhibitor of CDK5, as a titrator. So maybe it just takes away CDK5 from the active complex. Maybe this is the way how the activity of CDK5 is being controlled. Maybe. First of all, we need to know what this complex is doing. This is expressed in the adult brain, but there can be many different functions. Well, the breakthrough came from this experiment. And this was done by Junko Dajima and by Zach Wills, our collaborator from Michael Greenberg's lab. In this experiment, they took mouse neurons. Actually, I think this is a rat neuron. They put it in culture, and DIV means days in vitro. So as you culture these cells, initially they don't express cyclin E, but then later, after 11, 15, 17 days, cyclin E is induced. CDK2 goes down later because they're not proliferating, and CDK5 is expressed. What they noticed is that the cyclin E induction correlates with the time when those neurons are start forming synaptic connections. And then also they found that this is the time that the synaptic proteins are being induced. This is the presynaptic marker, and there's a postsynaptic marker. So can we conclude that cyclin E plays some role in synapse formation based on such a strong correlation? What do you think? You think yes. I think no. This is just a correlation. maybe you could do another experiment. You could correlate with something else, right? So the fact that cyclin E is induced at a time when the synapse forms, well, that's consistent, but it doesn't mean that this is that this is linked, right? Since I came to St. Petersburg, it's very cold, but of course it's a pure coincidence, hopefully, right? So the same could be here. So how can we address it? How could we kind of nail it to show that cyclin E plays a role in synapse formation? Sorry. Knockout, right? So the knockout is the way to go, and then you know that we have a conditional cyclin E knockout mouse. This at that time we didn't have it when I had a first talk, and Yang Geng has made it, and there are two E-type cyclins, and I told you that the E2 knockout has virtually no phenotype, so we have conditional cyclin E1. And E2 is knocked out, but it doesn't really matter. And then when we put Cre here, we're going to make this cell cyclin E knockout. So now we do the following experiment. We take these mice, cyclin E is expressed, so these are conditional knockout, everything is cyclin E is expressed. We put the neurons in culture, and then we electroporate them with the Cre recombinase. But of course we need to see which cell got the Cre recombinase, so we also co-electroporate with GFP. So the cells that are electroporated become green, and then they're also deleting cyclin E. And then we are going to stain these cells for the presynaptic marker and postsynaptic marker. And we are going to look for colocalization. If we see that the same place stains for presynaptic marker and postsynaptic marker, we call it a puncta, and we call it probably a synapse. So then we count it. So this is what happens here. These are the puncta in before, and this is after ablation of cyclin E, and this is quantification. This is number of synaptic puncta before, and this is after knockout of cyclin E. Once you get rid of cyclin E, the number of synapses goes down almost twofold. But I was not being neurobiologist. I was not very impressed by the staining, so we collaborated with my friend's lab, Steve Moss from Tufts, where they can actually stick electrodes and measure the synaptic potential or electric currents in the neurons. So so each spikes correspond to the postsynaptic currents. This is before, and this is after ablation of cyclin E. So as you can see, when we turn off cyclin E, that the amplitude goes down. This is amplitude from here to here, and this is smaller from here to here, and then the inter-eval frequency goes up, meaning they are more and more rare. So this slide actually convinced me that cyclin E has something to do completely independently of the cell cycle, because here you can take the neurons that are non-dividing, they have not been dividing for 2 weeks, they are terminally differentiated, you turn off cell cycle protein, and now you can see electrophysiological differences. So I think that this convincingly, it is so convincing for me that cyclin E plays a role in synaptic function, because if you turn off cyclin E in proliferating cells, that the electrophysiological status of the cells, the synaptic current changes. But of course, it doesn't tell us that it has anything to do with CDK5, right? This could be completely independent function. So how can we tie it up with CDK5? How would you do it? It could be that this is some completely independent. We discovered something new about cyclin E, but it has nothing to do with CDK5. Of course, we have found that cyclin E forms almost a stoichiometric complex with CDK5, so we can make the hand-waving argument and you say this is the major partner, so it must be dependent. But is there any way that we could do it directly? Do you have any suggestions for me? Okay, so this is our model. So maybe you can help me. Maybe we can do it together. So this is our model. And our model that we proposed is that CDK5 can bind P35. Of course, this is not our model, this is textbook knowledge. And P35 activates CDK5. And now we are proposing that cyclin E binds CDK5 and inactivates this. So, if cyclin E binds and inactivates CDK5, then when we knock out cyclin E, the CDK5 becomes hyperactivated, right? Because we were proposing that cyclin E is titrating. So if we knock out cyclin E, we hyperactivate CDK5 and we have less synapse. What would you expect if our model is correct? What would you expect if you overexpress CDK5? What would happen? Sorry? The same thing, exactly. So that's exactly what we postulated. We said if cyclin E inhibits CDK5 and if cyclin E knockout hyperactivates CDK5, let's just hyperactivate CDK5 and see what happens. So here we take the neurons and either we knock out cyclin E, and as you can see the number of synapses goes down as I told you, but now we take the wild type neurons and overexpress cyclin overexpress CDK5, hyperactivate CDK5, and exactly as you predicted, there's the same phenotype. So far so good. We can mimic the phenotype of cyclin E1 knockout with CDK5 overexpression. Well, the second kind of thing we said is that we said if we knock out cyclin E, we hyperactivate CDK5. What would happen if we knock out cyclin E and at the same time knock down CDK5? What would happen, do you think? Yeah, so the expectation is that it should be back to normal, right? That is, do we all understand? Because we are proposing the knockout cyclin E hyperactivates CDK5, and that's what those bad things happen. So let's knock out cyclin E and then knock down CDK5 and see what happens. So this is number of synapses before, this is after the knockout of cyclin E, and then we knock out cyclin E and we knock down CDK5, and we can restore back the number of synapses. So this was all done in vitro cultured neurons. But of course, the neurons in our brains not function in a single cells, but rather in the neuronal circuits. So we wanted to know what happens if we get rid of cyclin E in the context of neuronal circuit. So the way the experiment is done is that we take a brain of a mouse, non-culture, we make a slice from the hippocampus, we put the slice immediately straight from the brain in vitro, and then with the GeneCan technology we electroporate single neurons with GFP to make them green and with Cree. And now we are going to see what happened to these neurons, but we are not going to see the synapses, although you could do this, but we are saying here for the dendritic spines. The dendritic spines are the postsynaptic structures where this is the anatomical place where your memory are stored. So there are very nice papers to show that as you learn, you are forming a new dendritic spines. If you remember this lecture, this is because you found the new dendritic spines in your brain. The bad news is that in order to, which is of course we all know, but this neurobiologists have also shown that as you make the new dendritic spines, you lose the old dendritic spines. So in order to remember this talk, you'll forget something else. Hopefully not so important, right? But you never know. So, but that's what happens. So here we are looking at the number of dendritic spines in control and in knockout mice. These are three different areas of hippocampus: control, knockout, control, knockout. So this is lower. And now we're going to knock out cyclin E, as here, but at the same time electroporate the same neuron with siRNA against CDK5. So we're going to bring down CDK5 and see whether we can rescue this phenotype. And sure enough we can. This is control. We knock out cyclin E, and now we knock out cyclin E and knock down CDK5. And now we can bring it back. So we felt that the most stringent way of testing this model would be to do as follows. We knock out cyclin E, and then if CDK5 is hyperactivated, then what do you think is going to happen to the substrate of CDK5? To the phosphorylation status? Sorry? Increased, right. And then luckily there are several synaptic proteins that are being phosphorylated by CDK5, and there are actually antibodies available against the CDK5 specific sites. So we can knock out cyclin E in the brain, and then we are going to look at this protein, and use the antibodies against these phosphosites, which are known to be phosphorylated by CDK5. So we take our conditional knockout mouse, and then we cross it with the nesting Cree mouse. This is a different kind of Cree, so I just want to spend 1 minute explaining to you. So far, all the Cree expression I told you was inducible one, the one that we give mice something, we turn on Cree, and then we delete our gene. Nesting Cree is a different kind of Cree. This is the Cree that is expressed in the brain. So it's going to you're going to make a mouse. Well, only the brain is going to delete cyclin E, and this actually shown here. So we have a mice running around which have either control brains, control mice have control brains with good level of cyclin E, and then our conditional knockout will return of cyclin E or delete cyclin E specifically on the brain. Of course, with such a developmental thing, first of all, you need to check, well, maybe this is knocking out cyclin E in the brain is going to do something terrible to the brain, right? I told you that cyclin E in the first talk is not required for development of anything except for heart and placenta, but we need to, since we are now talking in the brain, we had to check this, so we did a very careful analysis together with our neuroanatomical and neurobiologists, and there's no phenotype. So like I told you in other compartments, cyclin E is not needed for brain development. So now we can look at the phosphorylation of those substrates, and this is using the phosphor-specific antibody. So this is protein called WAVE, this is phosphorylation in a control brain, and this is cyclin E knockout brain. It's clearly higher. This is another site. This is control and cyclin E knockout, clearly higher. The level of the protein is not different, just the phosphorylation is different. This is another CDK5 substrate, synapsin. This is control phosphorylation. This is cyclin E knockout phosphorylation. The overall level does not change. Cyclin E is deleted here. CDK5 is similar in both of them, but here CDK5 is hyperactivated. So now we know that in the brains of cyclin E knockouts, some proteins are hyperphosphorylated. So we have a biochemical phenotype. We have something abnormal that is at the molecular level. But we want to see some actual real abnormalities at the phenotypic levels. So first of all, this phosphorylation of the CDK5 of WAVE is very important. CDK5 phosphorylation inactivates WAVE, and WAVE is required for formation of dendritic spines. So if you hyperactivate CDK5, you hyperphosphorylate WAVE, as you can see here. We are expecting to decrease the activity of WAVE and to decrease those dendritic spine maturation, and that's indeed the case. Here we are looking at the size of dendritic spines as a marker of maturation. These are the dendritic spines in control, and these are in cyclin E knockouts, and this is the quantification. I told you that this is the place where the memories are stored. So hypothesis here, please remember this, we'll get back to later, is that probably these mice, at least this is consistent with these mice not being able to store their memories correctly. The second thing is that we look at the synapse size, and when you look at those electron micrographs, this is control, this is knockout. It's not very obvious, but when you measure hundreds and quantify, there's a difference. And also, this is a bit complicated slide, but I walk you through because I think the take-home message is important, is that CDK5 indirectly inhibits the kinase, protein kinase A. And protein kinase A, or PKA, phosphorylates a protein called NR1, which is the glutamate receptor. It's a very important receptor in our brain that is required for learning and memory. And this phosphorylation is required for synaptic localization of this protein. So if CDK5 is hyperactivated, then this protein is going to be super inhibited. There is going to be less phosphorylation of NR1, and there is going to be less synaptic localization of this protein. And that's indeed the case. This is the phosphorylation of this subunit, of glutamate receptor control and cyclin E knockout. This is lower, and this is quantification, and this is synaptic localization, control, and cyclin E knockout. Now the localization of this protein is very important because this is required for NMDA currents, which are needed for learning and memory. So now let's check the NMDA currents. We are going to stick electrodes into the brains of the mice. So this is not in vitro culture cells. These are the brain, and then the postsynaptic currents are those lines 1, 2, 3, 4, and you can see this is 1, 2, 3, 4. There is much more. The frequency goes down, and in particular, when you look at the NMDA currents, this is the glutamate current. This is the control, and this is cyclin E knockout. So those currents are reduced. So we not only have the biochemical difference, but we also have electrophysiological difference, as I showed here, and also structural, because we see that there's less dendritic spines and the synapse is smaller. So this suggests that cyclin E, that cyclin E is required for forming the memories, right? Because all those lower NMDA currents, the smaller dendritic spines point that cyclin E is required for learning and memory. And if we don't have cyclin E, then these mice should not remember very well. So we can actually test this. There are two ways of testing this. This is the electrophysiological way, and the way we do it is that we measure the electrophysiological currents in the brains, and then we stimulate them with the electric current. So the longer the stimulus, the stronger the stimulus, then the longer the memory. It's the same in humans, right? If I give you an okay talk, it's going to be a weak stimulus, and probably after a week you will forget it. If I give you a fantastic talk, then you will remember for the whole year. But if for example, I suddenly drop dead, this is a super strong stimulus, right? This never happens. So this you'll remember till the end of your life, right? If you're very old, you will say, this guy came from America and he dropped dead. So hopefully will not going to happen. So the stronger the stimulus, right? The stronger the shock, then kind of the longer you're going to remember. And it's similar in the mice when we give them the electric stimulus, and then you get the synaptic transmission which goes down, but it's still over the threshold for 60 minutes. This is long-term potentiation. So in mice, 60 minutes is long-term. Hopefully you remember my talk longer. And then in the knockouts, on the other hand, this long-term potentiation is decreased right from the outset, and then the synaptic activity is lower. So this suggests, this electrophysiological correlate of learning memory, that they cannot form memories, because this the persistent synaptic activity is the sign of remembering. We can test the memory more directly, and this is done here. So in this test, we put two different objects into the cage, and then we let the mice explore those objects, and then we come and we move this object here. So we do it like this, and now we measure the time that the mouse spends kind of running around this object versus this object. So a control mouse, as see here, will spend less time in the old object and more time in the displaced object, because this is something new. So the mouse remembers that this place, that this thing was in different place, and now mouse is exploring this one. But in knockout mouse, so this is this brain-specific knockout, they spend exactly the same time in the old versus in the new one, because they don't remember where it was. Of course, like with everything, this is open to other interpretation. There are things, maybe these mice are lazy, right? Maybe these mice are not interested. Maybe these mice don't have novelty seeking. So with electrophysiological thing, it's clear. You see high current, low current, but it's not so biologically relevant. Here you see there is a different behavior, but there could be different ways. And therefore we do another test. And in this test, this is a water maze test when we throw the mice into the water. And of course, mice don't like to be in the water, and we are nice people, so we are providing them with a platform. And then the pool is circular, but just for you, I'm just dividing them into quadrants. And the northeastern quadrant, where is the platform? And the mice can see the platform, so they swim to this platform, and they go out. And then, after several trials, we submerge the platform. So the platform is still there, but mice cannot see it, but hopefully they'll remember where the platform was. And then we measure the time, so we throw the mice in different places to confuse them even more, and then we see what percentage they spend in this quadrant searching versus in this quadrant versus in this quadrant. So control mice, shown here in white, spend most of the time in the northeastern quadrant and relatively little in other quadrants, because they remember that the platform was there. But knockout mice cannot remember. Once you submerge the platform, they are shown in black, they spend exactly the same time in each quadrant, because they cannot form memories that the platform was there. And lastly, we are going to test which is called cognitive flexibility, but I think it's intelligence. I think that actually you need to have in order to be successful scientists, meaning not just to learn, and I really stress that learning is also important, but rather relearning. In other words, not being stuck by one thing, but realizing that this information is no longer useful. And then forgetting something, or maybe forgetting but ignoring some old and useless information. So now we're asking mice to ignore it. So experiments are as follows. We put the platform here, and then we teach the mice that the platform is there until the most stupid mouse remembers that the platform is in this quadrant. And now we are going to put the platform to the new quadrant. And we are going to throw the mice into the water and see how long will it take them to figure out that what they learned no longer applies. And the way we do it is that we throw them first time, second, third, fourth, fifth, sixth to 12 times, and we measure how long does it take them, how many seconds does it take them. Of course, they are going to look here, but how long does it take them to realize that the platform is somewhere here. So in white are control mice. The first time you throw them, it takes them 30 seconds to find the platform. The second time, it's even worse, 36. But already the third time, it's close to 20, and by the fourth time, they find quite quickly, because they remember that there is no point searching here. But our mice, shown in black, they really cannot learn anything. Look, this is first, second, third, fourth, fifth, sixth, seventh, eighth. Well, maybe by the seventh trial, they finally get it, that the platform is not here, but it still takes them substantially longer than control mice. So that's it. So in summary, I told you about a new function about cyclin E in the brain, in non-proliferating tissue, and I mentioned you that cyclin E binds CDK5 and inhibits its activity, and therefore CDK5 can be either an active form together with a classical activator P35 or together with cyclin E. In this way, cyclin E controls the phosphorylation of those synaptic proteins. One thing we are now trying to understand is we want to know, number one, do the level of cyclin E change during our learning? Number two, obviously some of us have better memory, some of us, like myself, has very bad memory. Maybe I can blame low level of cyclin E in my brain. There's one possibility, but seriously, there are people with very severe learning disabilities, and we want to know whether the abnormal level of cyclin E could underlie those learning abilities. And lastly, we want to know whether the levels of cyclin E could underlie some neurodegenerative diseases like Parkinson's disease and Alzheimer, because they originate from cells which express high level of cyclin E. This was done by the graduate student, and this is a mouse. Cyclin E, and it can travel its known route, the cell cycle route, by activating CDK2 and CDK1, but there's also a new function by inactivating CDK2. And lastly, I want to close this part by saying that the cell cycle machinery was worked out in yeast. And I think somebody asked me about this transcription factor, and we were talking about this is yeast transcription factor, but this is the cell cycle machinery in yeast. And this is the transcription, this is the cell cycle machinery in mammalian cells. And one thing that became obvious at the last year of the previous century is that those cell cycle machinery is organized in a very similar way. The names are different, but the proteins function in a very similar way. Moreover, you can take, for example, you can remove this gene from yeast, put human protein, and human protein is going to drive cell cycle in yeast, almost as good as a yeast protein. So this is amazing. So those billion years of evolution, this machinery is so conserved. It works the same way, and you can take one protein, put into one species, and still, so those billion years of evolution, they are going to function. But one thing that we are now discovering is that in the process of evolution, yes, the core cell cycle machinery has been discovered, but in our cells, in the evolution, those core cell cycle proteins starting acquiring new functions that have nothing to do with the cell cycle progression. And today, I told you about the function in the brain. Tomorrow, I'm going to tell you about also completely non-cell cycle related function of cell cycle proteins that they required through this journey from yeast to our cells. In this case, somewhere billions of years ago, in the brain of our ancestors, cyclin E, which is normally silenced in non-proliferating cells, probably became de-repressed. By being expressed in the neurons, cyclin E wants to bind cyclin-dependent kinase, CDK2 or CDK1, that's the preferred partner. But in brain, there is no CDK1 and CDK2. Instead, the only good partner that it could bind is a similar kinase called CDK5. But because of the different structure, Sicinski cannot activate CDK5. It then activates CDK5. So it acts as a buffer against, in this way, so this accident of evolution, our memory forming ability acquired kind of a new layer of control, new ways how we can control our memories, but being able of this key kinase to be regulated by another protein that by yeast cells is used, and by cancer cells is to use cell cycle progression. So this is Junko Dajima, a person who drove this project. Yang Geng made somehow it happens that all the stories that I tell you are made by females and Asian females. But you know, it's in the next talk, you know, just to not to be sexist, I'm going to tell you the story of two guys actually, of a French postdoc and of a Thai postdoc. So I don't appear like kind of being a sexist and only promoting women in science. Anyway, so but here we have only women. So Yang Geng made this knockout mice. Sylwia Gawczak was a student from Poland who was helping Junko Dajima. Marie Gercois was a former technician. Now she's a graduate student from Haiti. Zach Wells, so he's a guy. He's from Michael Greenberg's lab, and he worked with Junko on this project. And then we got great help from Stephen Moss's lab, Michał Teronuma, Karla Kreczmanova, and Michał is from Japan, and Karla is from Czech Republic, and Tariq Dib is American, and then Yasmin Dassa Kolday from Senegal in Gerald's Martin lab helped us with this proteomic approach. And this is our lab a year ago. Maybe I'll spend a minute here just to emphasize how international science is in the United States. So Yoon is a person whom I talked yesterday, the person who did the shutdown of cyclin D1 and D3. She's from Korea. Li Jun is from China. Nan is from China. She's from the United States. She's from the United States, China. Junko from Japan. Lisa Bex is American. This is Sylwia Gawczak. She's Polish, but 50% Latin American. This is Per Hydrik, which is Swedish. This is Darek Górecki, Polish. That's me. That's Siwanon. This is a guy from Thailand. You will hear about his talk tomorrow. Sheila is from Brazil. Marie is from Haiti. Was, because she's now a student. Wojtek is from Poland. Leah is from Germany. Joasha is a former student from Poland. Yang Geng is Chinese. I talked about her work today. Conditional knockout. This is Tobias Otter from Germany. Nali from China. Lars Anders from Germany. And this is Xiao Yu from China. Okay, so I stop here and take questions.